Our topic review is about microfluidics devices for extraction and detecting airborne particles and aerosols by Shan Abdulkadir, Esteban Espinoza, and Dan Nguyen for our microfluidic fabrication and design class by Professor Sang Jun Lee for spring 2022. So first of all, what are airborne particles? Airborne particles are a complex mixture of solid and liquid droplets that remain suspended in the air. They can be of non-biological and biological origin, and we monitor them because we want to assess risk and hazards like pollution monitoring as the air quality decreases and pathogen detection.
A study by Passy et al. On subway passengers' exposure to airborne aerosol particles and bioaerosol particles in the respiratory tract found that the total concentration of bacteria exposure was 7 times higher than the specified exposure limit. Prolonged exposure to these pathogens can cause health issues such as diseases and immunological disorders. The fact that COVID-19 can be passed through bioaerosols is another important reason to monitor them. Even if no pathogens are present, other particles in the air can cause issues as well. Ardon Dreyer found that exposure to high concentrations of dust can lead to the likelihood that epithelial cells will die and decrease cell viability.
This microfluidic chip is extracting microorganisms directly from the gas stream, and unlike most microfluidic chips that uses liquid droplets, this chip uses gaseous state for capture and extraction and uses inertial principle of the movement of microenvironment particles for separation. The chip consists of 2 stages. The first stage is coupling of parallel double sheet focusing, and its function is forcing the particles to concentrate as it flows through the center line of the microchannel. The second stage is radial sheet flow acceleration, which adjusts the distribution of particles based on size and mass in the capture zone and will decide its capture area. Even though the driving force of moving the particles is airflow, this chip will still use active flow in focusing and in separation that will become active. Now we will talk about a digital microfluidic for detecting of selected inorganic ions in aerosols. This microfluidic device uses digital microfluidic technology, aerosol impaction, and chemical detection in the same chip, and targeting specific inorganic aerosol components, which are sulfate, ammonium, and nitrate. First, the digital microfluidic technology uses electrowetting. The microfluidic consists of a top and a bottom layer of electrode with an insulating layer deposit over the electrodes and a fluid between layers. Second, the aerosol impaction. The lower of the chip is used as a collecting flat surface, while the upper plate is used for air to flow, and as it flows into the microchannel and impacting the lower plate, the air then goes parallel to the lower plate. However, the flow does not affect the microdroplets. Thirdly is the chemical detection, which uses colorimetric analysis, where it will target the analyte and mix it with a color reagent to change its color that will indicate the concentration.
Another method of airborne particle extraction is groove-induced envelope or grip flow. The purpose of grip flow is to focus the flow towards an outlet port. As seen at the arrow labeled Roman numeral 1, the main flow channel is focused and accelerated by the channel that becomes more narrow as it goes on. This focus is supplemented by 2 side channels next to the main flow, which allow for better horizontal focus. The grooves seen here introduce a pressure gradient perpendicular to the flow, establishing a velocity field able to rearrange the airflow within the channels. Another method of extraction for airborne particles is herringbone-based microfluidic chips, which are primarily used for the collection of bioaerosols. First, a pump is used to continuously suck the fluid into the chip containing the staggered herringbone structures, depicted in image 2 on the left. These structures allow for a chaotic vortex of the fluid, causing the bioaerosols to rotate and stretch. This motion greatly increases the chances of contact between the particles and the walls of the microchannels. This contact results in the particles sticking to walls, allowing them to be collected later on.
So there are 3 applications that are introduced in this presentation. On the left-hand side, the first application is a 3D functional human microvascular network in a microfluidic device. This one creates human vascular endothelial cells to form tissues like microvessels that has a behavior that is similarly to human blood vessels. The middle one is the handle COVID-19 particle captured by a paper microfluidic chip. Smartphone-based paper microfluidic chips can detect COVID-19 particles within 30 minutes. The apparatus seen in part A of the center image was made to simulate a person clogging 2 times in a room the size of a typical classroom with air conditioning. The third one on the right is the application of monitoring of airborne particles is the effective density monitoring of hazardous nanoparticles. While engineering nanoparticles are becoming widely used due to their unique properties, they are also becoming a cause of concern with regard for the safety and biological effects. And for the example, The microfluidic chip is able to characterize the nanoparticles in terms of two characteristics, diameters, by applying the electric charge to them. For the conclusion, in this presentation, we discussed about the four methods, that is growth-induced envelope flow, herringbone-based microfluidic chips, a prototype aerosol detection system, and high precision extraction and concentration detection of airborne disease microorganisms. And we also discussed about the three applications of this one is the 3D functional human microvascular network, handle COVID-19 particle capture by paper microfluidic chip, and effective density monitoring of health hazardous nanoparticles. And this one is the reference of our presentation.